Okay, so here we are back talking about the muscle twitch, which again is basically the stimulation of a muscle contraction with just one action potential being sent to it. All right, and we talked about how the latent period was basically from the time the motor end plate is depolarized because the neurotransmitters have been released and they go to the chemically gated channels, open up, allow sodium to rush in, a little bit of potassium to leave, mostly sodium rushing in, and that depolarizes, which then depolarizes the inside of the cell and then depolarizes the sarcolemma. So when that sarcolemma is depolarized and the action potential starts across the sarcolemma and then down the T tubules, which are surrounded by the terminal cisternae, then what that's going to do, that's going to cause calcium to be released, of course. Then when calcium's being released, it goes actually into the myofibril, goes to the troponin, which causes the tropomyosin thread to be shifted off the binding site, allowing the myosin crossbridge to grab it. Now, all that happened in this extremely 5,000ths of a second time period called the latent period. And then, of course, our next period is the contraction, which means now the myosin crossbridges grab and actually pivot and pull the myosin, the myosin crossbridges pull the actin, and then our sarcomere starts shortening, meaning the H zone also shortens. Okay, now that's all happening during the contraction. Well, relaxation, we know what that means intuitively, but what that means as far as this chart is actually we start pumping the calcium back into the sarcoplasmic reticulum. Then our myosin cross bridges quit having the ability to pull, but instead they're released. And when they are released, then the actin molecules start sliding back. Our H zone increases, and our relaxation time occurs, which is the longest of these periods of time, but still maybe 20, 40, 55 milliseconds, so a little over half the whole amount of time that we go into relaxation. But of course, what we want to talk about now is how do we go from this twitch that we see here to a full muscle contraction, which we would experience for most types of movements or most things that are going to hold something in a stable position. Okay, so again, muscles are when they contract, they're either moving something or they're stabilizing something. Okay, now, so let's start looking at the next graphs and see what happens, because remember, this happened due to one stimulus, one neurotransmitter releasing its, having one action potential, releasing the neurotransmitters at the neuromuscular junction one time. Now, if we look at the graph now, we see it looks different than the graph we were looking at earlier. The graph we were looking at earlier had the latent period, it had the contraction, and a full contraction period. But notice here, what I have is I have a contraction, I have a partial relaxation, but then I have another contraction, which is even larger, and then I have a full relaxation. So what this is representing is, instead of me stimulating it or sending just one action potential, what if I sent a multiple, or in this case, a double action potential? What if I sent an action potential, and before I allowed that to totally relax, I sent another action potential? Then notice now I have kind of a stair-step effect on this, and this is actually called a temporal or wave summation. In other words, if I stimulate more than once and within a certain time period, then I actually get a more full contraction. So let's look at this again. This is again temporal summation of two stimuli. So again, a twitch was only one stimulus. Now we have two stimuli, and we stimulated it before there was a full contraction.